allergies. Today, Dr. Patel is going to talk about specifically how understanding how peanut-specific antibodies evolve in food allergic patients um, undergoing treatments. So please, let's welcome Dr. Patel.
Thank you for that very kind introduction, Chris. Talking about the future of antibodies and food allergy is one of my very favorite things to do. So thank you for letting me share some of that with you all today. Our lab is fortunate to be part of FASI. FASI has been this wonderful community of unique minds that are brought together to decode the complexities of food allergy. And as a translational researcher, our lab has particularly benefited from hearing about cutting edge science from a variety of disciplines, inspiring us to think and approach our own work in new and interesting ways. And while our work for FASI is really focused primarily on antibody evolution today, we're really going to take a broader look at some of our work and thoughts about the future of antibodies and food allergy. So antibodies are one of the most powerful effector molecules in the body. And while we've known about antibodies for a very long time, the past decade has been monumental for antibodies. Never before have we been able to decode and harness these molecules to treat human diseases. For the past 2 years, we have all lived through this incredibly unique pandemic due to COVID-19 that really illustrates the power of antibodies, the central role of the adaptive immunity in mediating clinical protection from foreign substances has been headline news. COVID antibody levels are now dinner conversation. We are now armed with clinical tools, vaccines to induce immunity, and a range of COVID-19 therapeutic antibodies to provide clinical protection from COVID. And even in these past 2 years, as the pandemic has reminded us about the power of antibodies, again, the protective power when induced by vaccines and therapeutic power when given to infected patients, antibodies have the ability to help us diagnose disease, treat it, and understand how diseases develop. And we hope that we can bring these insights to the field of food allergy. So a lot of the antibodies' ability to do their many functions are due in part to the way that they're structured. Antibodies are Y-shaped. The two arms at the top bind to antigens. These arms have finger-like areas, which can be many different variations that allow them to recognize or see many different kinds of antigens, including allergens. And on the bottom, the stalk region that binds to receptors on the surface of cells, these stalks come in distinct subtypes, such as IgA or IgG and IgE, just to name a few. And these subtypes can bind to different receptors on different cells and have different kinds of functions. Today, we're going to talk about food allergies that are caused by IgE or allergy antibodies, those antibodies that really drive allergic reactions, but we'll also focus on IgG antibodies. So these IgG antibodies that might be able to bind to allergens to block these allergy IgE antibodies from activating the allergic response. In food allergy, exposure to the allergen causes activation of allergy cells by IgE. So allergy cells, for example, like basophils in the blood or mast cells throughout the body, these cells have receptors on their surface that bind to IgE. And when this IgE recognizes or binds to allergen, it can activate the allergy cell to release chemicals like histamine that result in a systemic allergic response or anaphylaxis that can involve the skin, the lungs, the gut, the heart and the blood vessels, and the brain. And this activation of allergy cells can be halted by epinephrine, which has been an important part of our ability to treat these kinds of life-threatening reactions. And so antibodies are really central to allergy. Their ability to recognize allergen drives the underlying disease, but we think that it may also hold the key to treatment. Immunotherapy has been used in the field of allergy for a very long time. We use incrementally increasing allergen exposure. And we've used this in a variety of different types of allergies, right? Food allergy being one of them. In oral immunotherapy for food allergy, we know that oral immunotherapy can increase the amount of allergen-specific antibodies. So those IgG antibodies that can recognize allergen in the blood of food allergic patients during treatment. But here we ran into a little bit of a conundrum. Namely, when we examined clinical trials of oral immunotherapy for peanut allergy, it seemed that everybody had an increase in the amount of allergen-specific IgG. It didn't seem to matter what kind of clinical outcome you had after immunotherapy. So in these two clinical trials, so food allergic patients underwent oral immunotherapy, and they were treated with high dose immunotherapy. And after the immunotherapy was stopped, some of them had sustained responses, meaning they were able to tolerate a high amount of peanut. But a lot of other patients only had transient responses. They started losing some of their tolerance. And both of these groups had a rise in the amount of IgG antibodies that could recognize peanut. And even if we really just focus down to the most relevant of peanut allergens, ARA-H2, we still saw that everyone who underwent this high dose immunotherapy had an increase in the amount of IgG4 antibodies, in this case, that recognize this immunodominant allergen, ARA-H2. And so we began to wonder why that was the case. And we began to wonder, what if it's not how many antibodies you have, but what kind of antibodies you have? What if it's not the quantity of antibodies, but quality? And we started to wonder whether those antibodies that were induced by immunotherapy in patients who had sustained responses were fundamentally different for those antibodies that might be induced in those that only have transient responses. We wondered about this because of something we already knew. We had already previously discovered that we can use allergy cells as kind of like a test tube, um, a biomarker, or a way of assessing allergy kind of in a little test tube, because these allergy cells, they have those allergy antibodies on their surface. And if we take them from patients undergoing oral immunotherapy, we stimulate them with ARA-H2, that peanut allergen, and we check how much they're activated, we saw that their activation falls over the course of immunotherapy. But more importantly, our group understood that actually their sensitivity to how much allergens seem to activate them also significantly decreases over the course of immunotherapy, but only in those patients who actually had a sustained response. And interestingly enough, this decrease in sensitivity of these allergy cells started early in immunotherapy, and it was persistent even after they stopped immunotherapy. And we knew that this decrease in sensitivity could be mediated by those allergy cells that were, by those allergy induced antibodies, those IgG antibodies. And so we started to wonder, is long-term tolerance associated maybe with some special antibodies are induced? Can those antibodies actually functionally suppress IgE activation of allergy cells? In other words, is it possible that peanut allergic patients treated with oral immunotherapy might develop specific protective antibodies that then sustains that clinical response to oral immunotherapy even after oral immunotherapy is over? And so before we could even get to answering that larger question, we really needed a deeper understanding of how antibodies that develop during oral immunotherapy can even recognize the allergen, and to connect that information to its functional protective ability. So we hypothesized that sustained responses to oral immunotherapy involve the induction of antibodies, IgG antibodies, that can more effectively block the allergen from actually being able to activate IgE and allergy cells, whereas those antibodies in transient response to immunotherapy, they actually don't do as good of a job blocking those allergens from coming in contact with the allergen IgE antibodies on the surface of these cells. And so we wanted to understand, how does the allergen and antibody interaction really drive that functional suppressive ability? We wanted to know, how do the antibodies recognize the allergen? Is it how strong they bind to the allergen? Is it where they bind to the allergen? Is it how many areas the antibodies bind to the allergen? And then understand how those different aspects of all of these antibodies are induced by immunotherapy really drives that clinical protection that we see in oral immunotherapy, particularly in those people who have sustained protection after immunotherapy. And so to study this, we utilized 2 clinical trials of peanut oral immunotherapy at MGH, where both children and adults underwent high-dose oral immunotherapy. And after finishing oral immunotherapy, they avoided allergen for 1 to 3 months. So we could assess whether they had a sustained response in blue or only a transient response in red. And from the blood of those individuals, we found the B cells that make the antibodies that recognize allergen. So we could actually take those individual cells, clone out their antibodies, and study them in the lab. And so we first began to understand how these antibodies bind to ARA-H2. We turned to our colleagues, and they were able to use X-ray crystallography to actually visualize where these antibodies bind to the allergen. So here we see the allergen in the middle in white, and we have the binding of two allergen-specific antibodies, the arms from one antibody in blue on the left and another, the arms from another antibody in yellow to the right. And the areas where these antibodies contact the allergen are colored in red and blue respectively. So using these types of methods, we could finally understand where these IgG antibodies could bind to the allergen, identify protective antibodies. And now we've also been able to show that these antibodies can actually block allergy IgE on allergy cells from getting activated. So now that we've kind of started to identify these protective antibodies, we want to understand why they develop in patients who have sustained responses as opposed to those that have transient responses. And part of this relies on understanding how antibodies in general develop. We spoke about how these little finger-like projections on the arms of the antibody actually recognize the allergen. Well, it turns out those variations that drive the ability of these antibodies to recognize allergen develop over a process of natural selection. And many years ago, we described that we thought that these antibodies and the way they recognize the allergen actually exist in many different patients, meaning that these are common ways that peanut allergic individuals have antibodies that can recognize the allergen. And that lets us do two things. One, it tells us a lot about how these antibodies are developing, that they can develop in so many people. But it also told us that the potential to develop protective antibodies might actually exist in a large number of peanut allergic individuals. And if we could understand how they develop in those that have sustained responses, we might be able to devise ways of inducing those protective antibodies to induce a long-lived, sustainable tolerance to food allergens in the future. So right now, we're on the cusp of gaining a deeper insight into the protection that antibodies can provide against IgE-mediated food allergies. By understanding how these antibodies see allergen and prevent allergen from activating allergy antibodies, we can open a new avenue for the development of both diagnostics and therapeutics for the treatment of food allergy. We hope this work will help us develop better biomarkers of tolerance by letting us look at when and how protective antibodies develop. This can help us personalize therapies. We can choose better suited therapeutic options, maybe more precisely prognosticate the outcomes of using particular therapies, and hopefully take some of that guesswork out of treating food allergies. But we also have this unique opportunity to innovate in this area, to develop new antibody-based therapies, such as providing antibody-based protection to allergic individuals. Our knowledge will help us understand not only what makes an allergen allergenic, but also how we might be able to better modify allergens. Finally, we might be able to shape antibody development to create a sustainable and durable antibody response in allergic individuals to get closer to the cure that we all seek for food allergy. And while we're excited about the work that we have done, we're even more excited to get started on the things that we've only been able to dream about until now. The support of FASI, a group of such diverse talents, has been a true gift. Thank you for your time. And FASI, thank you for the support.